Good morning, good afternoon, and good evening to everyone who dialed in. Welcome to ACE, America with China Empowerment Seminar Series. I'm the moderator for today's webinar. My name is Xuedong Dai, the Executive VP and Head of IDSU at Medicilon. First of all, please allow me to introduce Dr. Chunling Chen, the founder and CEO of Medicilon, to give us the opening speech. Dr. Chen obtained his Master of Pharmacy degree from China Pharmaceutical University and his PhD degree in pharmacology and toxicology from Oklahoma State University. He returned to China in early 2004 to found Shanghai Medicilon Biopharmaceutical Company. Without further ado, let me welcome Dr. Chen for the ACE opening speech. Dr. Chen, the floor is yours.
Thank you, Dr. Tai. Happy Chinese New Year to everyone. So I would like, first of all, I would like to thank you several associations to support this, seminar, including SAPA, and CBA, Chinese Bioscience Association, BCIC, Boston Chinese Investment Club, and CABS, Chinese American Biopharmaceutical Society. The year of the rabbits, that's 2023, is going to be a great year, full of new challenge and opportunity. To better prepare us for this exciting year, Medicilon is rolling out the ACE seminar series. ACE seminar aim to bring together experts and executives in life science between China and the US to systematically introduce the fresh idea and the key element to share their expertise and conduct it depth of discussion. So I will quickly introduce our company. I'm a founder and CEO of this company. During the last 19 years, we're building up fully integrated drug discovery from idea to IND filing, including drug discovery for new chemical entity, then get into CMC development and the preclinical research, finally to submit the IND to China and US and other FDA entity. So last 19 years, we served over 1,700 clients around the world. Currently, we have over 3,200 employees, including chemists, biologists, preclinical scientists. We own over 80,000 square meter laboratory and facility internationally. I just give you our achievement. Just last year, that is 2022, we have our sponsors submit over 100 IND got approved in China, US, and Australia. As you can see this slide, 97 IND got approved in China, 11 got approved globally in US and Australia. Also, I'm glad to show you our facility in Shanghai. We have 4 facilities in Shanghai, and one facility is very close to Shanghai Disneyland. So we would like to welcome everybody coming back to Pudong Airport. Let's stop by us, then go to Shanghai Disney, only 10-15 minutes away from Pudong International Airport. Also, we have another facility in Hangzhou and Boston. Okay, so we also welcome everybody who is interested in joining us, work in China, US, especially Boston and other areas. So in first, ACE series, we are talking IP, because IP is critical for new drug discovery and development. And we have great experience and create one very special model Besides our traditional FTE service or non-FTE service or traditional service, we create called IP and plus CRO plus pharma to bridging together between China and the US. So therefore, IP become very critical. And just imagine without the CRO, it also have an opportunity to create a cooperation between US IP and the Chinese pharma together. Unfortunately, without the CRO, sometimes are going to become a challenge to get a deal done because culture, language, project evaluation, project validation, etc. So we are glad as one of the participants for overseas IP and the Chinese pharma working together using our professional validation platform, evaluation skill, and the dual filing capability. So CRO become like a Medicilon, become a very reliable bridge and a bond between China and US in terms of IP transition or licensing and license out. We already created one several case, very several case. As you can see from here, we have one Chinese company called Fenzen Pharma licensing compound from New Jersey, SKPP, using our IND enabling study for bring together these two companies working together. And we have other case like bridging Suzhou company and the Sweden company working together using our space. Therefore, we're thinking to working together, licensing, license out, IP protection is critical. So that's why our first series are talking about IP protection. So we would like, as the CRO play more and more important to do license between China and the US. Okay, that's what I want to thank you. Everybody attend this seminar and would like everybody pay attention IP protection and have more and more licensing and then license now between China and US. Thank you very much. Back to Dr. Dai.
Thank you, Dr. Chen. Let me share my screen. Thanks again, Dr. Chen. And can we see the speakers? Okay, today we're so glad to have Dr. Yang and also Dr. Felstein from Finnegan Law Firm to give us the kickoff seminar of our ACE seminar series titled Building a Strong Patent Portfolio to Gain Market Competitive Advantages. Before we get to their seminar, I would like to say a few words about myself and also Medicilon. So I received my PhD in organic chemistry and postdoc training at MIT. I then worked at US biopharma Xenoport and later at GSK and Johnson & Johnson on the small molecules for CNS, infectious disease, and oncology for more than 20 years. I have been involved in the development of the marketed drug Horizon for restless leg syndrome and several clinical candidate drugs such as J&J 7527-6617. I joined Medicilon 2 years ago as the Executive VP and the head of IDSU, International Discovery Service Unit. And I'll say more about this unit in the following few slides. So as Dr. Chen just mentioned, Medicilon is a one-stop shop platform for drug R&D services, which can help our clients bring their ideas all the way to IND filings. So Medicilon has three main divisions: drug discovery, drug development, preclinical research. And in drug discovery divisions, we have full-scale chemistry and full-scale biology, from protein production, in vitro pharmacology, all the way to compound screening. And in drug development, we have drug substance, API, and drug product formulation process chemistry. And in preclinical research, we have DMPK, pharmacology, efficacy models, safety testing, and toxicology. So IDSU is a unit sitting in the chemistry chemistry part of drug discovery division. The mission of IDSU is to form a synergistic partnership with our global pharmaceutical companies and offer our sponsors high quality, cost-effective design and executions of synthetic medicinal chemistry services and program delivery by our teams with world-class capabilities and scientific rigor. And also state-of-art laboratory instrumentation, chemistry platforms, and track record of innovation and success, and a broad worldwide network. So the IDSU sits in our Nanhui campus in the southern part of Shanghai. We offer traditional business models such as fee-for-service and full-time equivalent, FTE. And our working relationship with clients is very simple without claiming any intellectual property. And we have a high quality team. The senior leaders all have over 10 years of CRO experience. And also 40% of the team has master and PhD degree holders. And we have several platforms such as photo redox, electrochemistry, catalyst screening, ADC, ProTech. And also for the chemistry, we have SAR synthesis, reference compound synthesis, heterocyclic, carbohydrate, nucleoside, nucleotide, etc. So these are all our expertise in the synthesis. And in addition, we can also work with our sponsors on a risk-sharing milestone-based collaborations. So we can offer chemistry, biology, and PK, PD, CMC, preclinical, and all the way to IND submission. Examples such as Dr. Chen just shared. And these are all like milestone-based. And we have project management team which can make sure everything is on the timeline, such as delivery of the lead compound within up to like 6 months, and lead op up to 6 months, and also the candidate identification within 3 months, and the preclinical study and IND filing within 18 months. And one example of our collaboration is with Seegene Tech on the ALK inhibitor CGT-9475. So for this compound recently, like in July last year, we got the compound got the FDA approval to enter the clinical trial, which is a big milestone. And Medicilon completed a full set of preclinical research services, providing the technical support for this compound to achieve the dual reporting and dual approval between China and US. And now it's time to give the podium to our guests, Dr. Yang and Dr. Filstein. Welcome both to ACE Seminar Series. Could you please introduce yourself? Thanks.
Thank you so much.
Go ahead, Yi, please.
Go ahead, Mark, please.
Okay. So I'll just introduce myself and then I'll turn it back to Yi. I'm Mark Feldstein. I'm a partner at Finnegan Henderson. Here, my office is in Washington, DC, and Yi will tell you a little bit more about Finnegan. I've been a partner, I've been at Finnegan for my entire career, a little bit over
20 years practicing intellectual property, all aspects really from prosecuting patents, obtaining the patents from the patent office, to arguing them to appeal boards and litigating them. I mainly work in the pharmaceutical and biotech area, as well as also some straight chemistry work. My PhD from before I decided to be a lawyer was from the University of Pennsylvania in physical chemistry. And I spent a couple of years as a postdoctoral researcher making sensors to detect biological warfare agents for the Naval Research Laboratory in Washington, D.C. And I'm very pleased to be here. It's great to be with Medicilon, and I want to thank you for the invitation and turn to you, Yi.
Thank you, Mark. Sorry, thank you, Mark. And again, thank you, Dr. Dai and Dr. Chen, and thank you for the invitation. My name is Yi Yang. I got my PhD. I got my bachelor's degree in biology and a master's degree in biochemistry in China. And I came to the US, got my PhD in molecular biology from University of Kansas. And then I did a postdoc at Dana-Farber, Harvard Medical School, Dana-Farber Cancer Institute for a few years. And I decided to switch to law. Then I joined George Washington University for law school. And then I joined Finnegan in 2014. So I've been working with Mark for over 8 years and learned a lot from him. And I've done a lot of work with him, including prosecution, litigation, patent litigation, especially from pharmaceutical side, litigation and patent challenges and due diligence work, licensing. And we just cover a lot of different areas in IP law, and we provide a full service. And it's an honor to be here again. Thank you so much for the invitation. And maybe I should start with my deck.
Sure, please go ahead.
Yeah, thank you.
Thanks.
Can you see my screen?
Yes, very well.
Oh, sorry. All right. So today we're going to talk about a little bit about our experience and thoughts over how to build a strong patent portfolio for biopharmaceutical companies globally. It's a global like a prosecution strategy. First of all, a little intro about the firm Finnegan. Finnegan, we have a long history. The firm was founded in 1965, and we have opened 11 offices globally, including in Asia, Europe, and the United States. And our headquarters office is in Washington, D.C. We provide a lot of services in the IP law area and in the four main jurisdictions, including Europe, Germany, UK, and US. As you can see on the slide, we do starting from prosecution, getting patents for clients, to later defending the patents or challenging patents or transferring the rights in patents, licensing patents, due diligence, managing patents. Everything we do is listed here, and we provide a variety of services, also cover trade secrets, litigation, antitrust litigation as well. We have a robust biopharmaceutical life science practice. We have 145 professional patent attorneys in our groups, including majority of them have advanced degrees, including me and Mark, with PhDs and masters in science before they started their legal career. And we cover a lot of different technological fields and provide the strength as a prestigious IP firm in the US. We represent a lot of leading companies around the world, such as AbbVie, Eli Lilly, Otsuka, and many other names. We also represent small biotech startup companies and try to help them to develop their portfolio and help them to grow their business, expand their business to grow stronger. So it's including the Chinese companies as well. So today I'm going to cover high-level strategic patenting for biopharmaceutical clients and companies, and Mark will provide further practical tips on how to achieve strong patents during prosecution in the US. First of all, I wanted to quickly introduce patents versus trade secrets. Both are very interesting, important tools for a company to protect the information or invention that is information or invention. but there are some important differences in among the two. For example, not everything is patentable or patent eligible. You have to satisfy the patent law requirements and make sure the subject matter is eligible for patenting if you want to protect your inventions by patents. For trade secrets, the protection scope could be broader. It can cover everything that you want to keep in secret, including information. But the confidentiality period of time is kind of different for the two protections as well. For patents, once you file the patent application to the PTO, USPTO, eventually the patent will be published either as an application or as a real issued patent. Well, for trade secret, you can kind of, if you want, you can keep the confidentiality for the indefinite amount of time. And talking about expenses, it could take some cost to prosecute and apply for patents in the United States, including the filing fees and patent maintenance fees and attorney's fees when you're trying to pay the attorneys to get a patent for you, while the cost is lower for to keep trade secret among your own company. Protection term is different as well. For patents, usually the protection only lasts until the expiration date of the patent. And for trade secrets, again, as long as you can keep it as a secret without public disclosure, you can always maintain the protection. But the strength of protection are different too. Patents provide very strong protection of your invention, such that any competitor or any person or company who make and use your invention can be sued for infringing your patent rights. And it covers the scenario when a competitor buys your product from the market and go back and try to take it apart and reverse engineer your product. As long as they eventually make the product that covered by your claims, they still infringe. On the other hand, for trade secret, the application is limited because you can only sue when you find there's a unlawful breach of confidentiality agreement between you and some party, such that your competitor, if they can come back and reverse engineer your product, you cannot sue them for that because there's no breach of any contract. And the two kind of disputes are governed by different laws in the US. And the last different point is the ease of control the property, because they're both properties of the valuable properties for the company. For patents, usually it's easy for the employer company to ask inventor employees to assign the patent rights to the company when they join the company, so that when the employee leaves the company, the property, the IP stays with the company. While on the other hand, if you have trade secrets, then any person, any employee has a know-how of your trade secret. When they leave the company, it's harder for the company to track and control whether the former employee could intentionally or unintentionally disclose the trade secret to other parties. So that it's complicated there. That's why there are a lot of trade secret suits nowadays. This former employee problem. So a smart company will think about the pros and cons for both tools and decide which ones to adopt to protect any information or invention. Some companies now, they use both tools to protect a combination of the tools to protect different kind of inventions or information for different purposes. So I think, again, that balancing the pros and cons using combination of the two could be a smart approach. Now, we talk about in the previous slide, we talk about not everything can be patent eligible. In the US, there are some subject matters that cannot be eligible for patents. You cannot apply for patents to cover those, such as laws of nature. For example, if you wanted to patent some metabolism of a drug in your body, because the body can metabolize a drug, it's a natural process following natural law. You cannot patent that. Or natural phenomena or product of nature cannot be patent eligible. For example, if you wanted to claim and cover a naturally occurring genomic DNA or naturally occurring mRNA or naturally occurring antibodies or proteins that is existing naturally in human body, you cannot do that. And also you cannot patent abstract ideas. Usually those restrictions are not an issue for compound claims because compounds are usually synthesized in vitro. And they don't exist naturally in human bodies. They don't have this issue. But as I mentioned just now, examples I mentioned, biological products such as polynucleotides or proteins have the unique potential risk because they could exist in human bodies. So that's why when we're drafting claims for those biological products, it's smart or routine practice now to add claim language such as isolated, engineered, or synthesized before those antibody or biological product language. So that's one thing that you have to keep in mind when you're talking about claiming biological products. And there's a hot disputed area that is diagnostic claims. In the US, the courts usually are not very friendly to those claims because many of the times the diagnostic claim they claim the process of, first of all, judging metabolism of the drug in a human body, and then designing the dosing regimen based on the metabolism. Again, to the judges, that sound to be abstract idea or claiming natural law of nature. That's why you have to be very creative and be smart when you're trying to draft claims for diagnostic methods. Now we talk about we want to talk about during your different stages of R&D. What kind of claims can you try to draft and try to apply? For example, during your very early R&D stage, when you're screening for a new compound or screening for a new biological product, antibody, or even during the preclinical stage, once you have an idea about the lead compound or the most favorite polynucleotide or protein sequence you want to cover, I think it's time for you to start drafting claims and applying for claims covering a class of compounds and polynucleotides covering the ones you like. You can also try to draft the claims to cover the solid form of the compound, such as the different polymorph forms of the compound. And you can also try to draft claims to cover the functionality of the proteins or the polypeptides or structure of them, for example, a card construct, stuff like that, to make sure you have security of the composition first to claim rights to your composition of matter. And then later on, when you get to the NDA, IND submission stage, once you nail down your formulation process, your formulation and of your drug product, or your manufacturing process, or both, it's time to think about drafting claims to cover your novel formulations and novel manufacturing process. And it sometimes happens during the FDA approval stage, or even after FDA approval stage, the brand name companies keep modifying and optimizing and improving their formulations and manufacturing processes. It's always okay to keep continuation applications to claim the improvements of those kind of aspects of invention. And then when you move down to the phase clinical trial stage, once you move down to phase 2, phase 3 stage, once you have a protocol covering your design of your trial, your clinical trial, it's time to think about claiming method of treatment, even starting as a broad claiming a method of treating covering the indication you wanted to target, or you can draft claims to mimic the language in your protocol, for example, the target population of the patients. Or later, when you get more data from the phase 2, phase 3 study, you can sort of develop further claims for more specific dosing regimen, and also covering the results from your primary, secondary endpoints. So there, again, as you see, there's an evolvement of different type of claims you can try to draft during the entire development to cover all aspects of your inventions. Now we talk about the slide just now talked about timing of draft different claims. Now we're talking about the scope of the claims. So what's this proper scope for your patent protection? I think, we can make an analogy. It's that drafting a claim is just like building a picket fence to protect your core technology. Okay, so the scope has to at least cover your core technology, but always as a patent owner, we want it to be broader to cover more, right? Because for example, you want to make sure you not only cover your core technology, but also you cover all your current pipelines, but also upcoming pipelines and your further improvements. How to do that? you can have families of patents alive, and also always filing new continuation applications to continue claiming the improvements of your core technology to make it always expanding the scope little by little based on your new invention. And also your claim also have to be broad enough to cover a competitor's product or their patents. Because once, for example, even if you don't have a product on the market, your competitor has a product in the market, but somehow your patent covered their product, they will feel threatened by your patent because they don't want to infringe your patent. So they may come to you when they get to the market, they may come to you and ask for licensing and pay you to avoid infringement accusation from you. If you have a product on the market that is competing against your competitor, if you have a broad claim covering both products, it could be used as very valuable bargaining chips to start negotiation with a competitor for cross-licensing each other's patents. So, getting a broad enough claim is crucial for your business success of your business. Now, for biopharmaceutical patent claims, there are examples of very broad genus claims. Again, we prefer broad claims in general because it can give you a lot of advantages in the market. So, for example, broad genus claims for compounds could be very classic traditional claims reciting a common formula with different variations of the different groups on the compounds, such that the formula itself can cover millions of compounds. And another classic way of covering antibody, for example, is to just claim any antibody binding to a new target antigen. And that kind of claim can potentially also cover millions of antibodies. For platform technology, and it's a routine way to cover at least the backbone structure and elements that can be applied to many different transgenes and to many different therapeutic applications. So those are the traditional and classic, very broad claims that can be drafted to cover the biopharmaceutical inventions. But how broad can you go? There has to be a limit, because if it goes too broad, then you will introduce more prior art to make sure such that your patent loses novelty and becomes not inventive anymore, such that it becomes invalid. For example, if you make a human or humanized antibody and you're becoming greedy, now you say, I'm going to draft a claim to cover all antibodies against this target. So your claim now covers even mouse antibody, right? And in the literature, there are people published about mouse antibody against the same target 10 years ago. So now the judge will say, well, sorry, your claim covers the old stuff. So your claim is invalid because it's not novel. So you may lose your specification, which describes your invention. If you provide limited example, you may lose the validity by lacking written description support or enablement support. So if your specification does not support the inventor's possession of the entire scope of the claim, then according to the law, the patent is invalid. So you have to control the scope properly. And today I want to talk about some unique challenges for biopharmaceutical claims, talking about providing support for your claims in your specification. Under the law, the patent must disclose either a representative number of species of the entire genus, the broad scope of claims that covered by the claim, or it has to disclose the common structure features, again, possessed by all the species in the entire scope of the claim. And in many cases, when the claim is too broad and the specification provides too few examples, the court always announced that, I'm sorry, the patent is invalid because you failed to support your entire claim scope. One example is one of the cases, it's a very famous case, Abbey versus Janssen. The Federal Circuit determined that when the claim recites full-length human antibodies defined by a range of binding affinity to bind to human IL-12 and their neutralizing activity to human IL-12, the claim covers potentially millions of antibodies with different structures because, again, many antibodies with different sequences can fit into this criteria in the claim. However, the specification discloses over 200 antibody examples in their sequences, but the problem is the 200 antibodies are from the same clone. So the inventor made mutations on antibodies to make them a little bit just like each other, but they share a very high percentage of sequence similarity, 90% similarity of the sequences. So the court said, okay, it seems that you have a lot of examples, but look how similar they are, right? If you compare this small group with what is on the market, because there are competing antibodies in the market. If you compare the sequence of the 200 antibodies with the ones on the market, they only have 50% identity. It's very, very different. And the structure of those antibodies and these antibodies in the specification are very different too, which means the examples here, they're too similar to each other, but very different from what else could be covered by the claim. That's why they're not representative of all the antibodies that are covered by the claims. That's why the patent claim is invalid, because you failed to show you're entitled to the protection to all the antibodies in the claim. As another example, in the Amgen versus Sanofi case, another very famous case in the United States, the claim covers monoclonal antibodies binding to at least one of a list of recited epitopes on the antigen, which is PCSK9. And the court said, okay, so again, this is a very broad genus claim because it does not define any structure or any sequence of the antibody. It just tells you any antibody that can bind to particular amino acids on this antigen could be in the claim. And let's look at what the specification discloses. It discloses the name of the antigen. Oh, that's this PCSK9. That's the antigen. It also discloses the X-ray structures of two antibodies binding to the epitopes on this PCSK9. So at the district court level, the district court judge said, okay, I think, because it's a jury case. So in every single jury case, the judge has to tell the jury, instruct the jury, okay, this is the legal standard when you talk about determining whether the claim is supported by the spec, because you don't know anything about patent law, you're just jury. So let me tell you, what is the standard? The standard is, as long as the antibodies bind to the newly characterized antigen, describing the antigen itself is sufficient to support the antibodies in the claim. So that's what the district court judge said. The parties, so that's why based on this instruction, the jury said, okay, we find the specification describes the antigen that's sufficient to support the antibodies. So they found the patent not invalid for lack of written description support. When the case got appealed to the Federal Circuit said, no, that's not right. That's a wrong instruction to the jury. So describing a new antigen does not describe the antibodies bind to it. It's not true. So please remand the case to the district court. Please reopen the case, get a new trial, determine whether the specification describes sufficiently the claims. Eventually, the district court found, okay, describing two structures of two antibodies is insufficient to support the very broad claim covering so many antibodies. So that's why the patent was found, the claim it was found to be invalid for lack of support. As another example, in Juno versus Kite, the claim recites chimeric T cell receptor encoding nucleic acid polymer, pretty much CAR construct, comprising a CD3 zeta chain proportion, a specific co-stimulatory signaling region, and any binding element that interacts with any target, which means it potentially covers any SCFV fragments that can bind to any target. It's a very broad claim. On the other hand, the specification only describes two SCFV examples, each binding to a different antigen. One is PSMA antigen, one is CD19. So when at the court, the patent owner argued that, okay, all the SCFV fragments look similar, And we described two different examples which bind to two very different targets. So we provide sufficient support to support this claim. The court said, no, you're very wrong, because you didn't tell me what kind of SCFV fragment can bind to what kind of target. You never provide any guidance in the specification. And there's so many antibody fragment can be there binding to any target. It's just like extremely broad scope. Two examples is insufficient. So that's why this claim is invalid as well. Okay, now I move on to another legal requirement. You not only have to provide written description support for a claim, but also you have to enable a person's good art to make and use the full scope of your claim invention. Again, it's a full scope. We were using another example, the Amgen versus Sanofi case. Remember, we talked about the case just now, but that particular claim covers antibodies binding to at least one of the epitopes of the PCSK. So that claim has been invalidated. Now the rest of the claim is the narrow one because it binds to at least two of the recited epitopes. And the court found, okay, again, based on the example only showed how to make two antibodies binding to the recited epitopes. It does not enable a person's good art to make all the antibodies covered by your invention. So that's why it's not enabled. So now the interesting development now is that after this decision was made, now the case is appealed to the Supreme Court, and the Supreme Court just agreed to take another review of the case and potentially a major change of the legal standard sometime this year. So we're excited to watch out for more guidance from the Supreme Court about the legal standard. Another example is a use of compound example. So this, in the Wyeth case, the claim covers use of a compound in the rapamycin class for the treatment of a particular condition. And the court found this rapamycin class covers thousands of compounds. They're all in the same class. The specification only discloses using a particular compound in the class for the treatment. So, so the board said, the Federal Circuit said, well, this is not sufficient because using a compound for treatment is not that easy, right? And because it's less predictable than making the compound. Using the compound is a higher standard. And you only show me one of the thousands of compounds is used and it's successful use. I don't even know how you can make all the other compounds in the class work, especially using for the treatment. That's why describing use of a single compound cannot support or enable PUSA, a person's good art, to use a large class of compounds. So the bottom line is, high level summary is there are a lot of unique challenges of claiming, reciting broad claims in the biopharmaceutical industry, but there are still ways of to make sure you have very valid and strong patent. Now move on. I'm going to pass the notes to slide to Mark.
Okay, great. You could move forward one more. So I want to talk about actual concrete things to do in preparing your patent application with your patent attorney and working with them to get the patent out of the patent office. Because there's a big difference, there can be a big difference between having that piece of paper that is a patent and having a good piece of paper that's actually going to be long-term business value to protect your rights to keep others off the market from copying your invention. It's unfortunate that too many good inventions, too many good ideas end up kind of a weak patent where they miss something. They didn't do all the possible things that you can do to make a strong patent. And ultimately, a court said the patent's invalid, the patent's not infringed. And at the end of the day, it didn't have a competitive value. So there's no magic recipe to how to make a strong patent. You have to keep in mind all the things that Yi said, but every case is different. Every invention is different. And it's frankly an art informed by a lot of knowledge about how patents are challenged on what you do to make a good patent. But there are some common themes. There's some common things that often work very well for a patent owner, and I want to talk about a few of them. And you don't have to worry about the details of this slide. This is really just an outline of what I'm going to talk about. So I'm going to go through it kind of quickly. But when you're preparing your patent, you can say, I made a formulation. Or you can say, I made a formulation and it overcomes these great problems of being stable from fracture from when shipping, and the drug remains stable on storage for a long period of time, and it dissolves faster, increasing the bioavailability. It's better, I think, when you're thinking about your invention, to think about what problem did you solve by doing this invention. It's really nice to have a formulation, for example, that has ingredients A, B, and C, but it's not so compelling that, wow, you made something, wow, the judge should agree with you, wow, the jury should award you lots of money. And so figuring out, understanding what problem was there that I'm solving, and then telling that, we say at the top, the invention story, telling the invention story for why you have really contributed something valuable to the field. And there are things to include to do this. And we'll talk about them. It's comparative testing that you would want to include early, not late. You want to disclose the parameters and how you figured out that there was a problem that no one else recognized, and then you solved it. That's a big contribution. You want to point out that everyone in the art said, go this way, this is the way you go, and you went this way. You went the opposite direction. That's unexpected, that's not obvious. And then the last point here is about prophetic examples. You can put in examples that you have not done yet to support all the things that Yi said about having enough examples to support your invention. You can put in working examples are great. You know, if you've been in the clinic, if you've been in animal studies, it's great. But you can draft hypothetical examples too to support your application. You go forward, Yi. So we're going to go through a couple of examples. This is one example where it was a formulation, eye drops basically, and they were able to make Senju was able to make a very stable formulation of their drug, and they claimed it, and it was very, very similar to the prior art. And they knew when they were drafting their application that what they're making, their formulation is very similar to the prior art. And they knew that they're going to have to show something special over the prior art. And so what some people do is they just wait and they'll get a rejection, they'll do their best with it. Senju took a very proactive approach. They did comparative testing in the first place, and presumably that is how they ended up with their formulation. But their formulation includes the component I have highlighted in blue, tyloxpol, and that's required. And they did comparison. They included in the specification comparing it to an otherwise identical formulation, but with polysorbate 80 in there. And what they found, and they told the patent office from the very beginning, is that if you make this one change, you increase the stability, as they measured it, from 51 to 73%. You basically increased it by 50% the stability. They put that story, they showed that what they had is not just a laundry list of inventions, but they had something that had inventive value, created a benefit for inventive benefit, a scientific benefit, a pharmaceutical benefit. And they put that in there, they got the patent. And then when Lupin, the generic company, challenged the patent and wanted to invalidate the patent later, the court, in this case, is the PTAB we're referring it to, they went back to the data in the patent and said, that is unexpected, that is non-obvious. And they had that story from the very start, and that made a strong patent. Next slide, please. So here's another one. Stability, as you'll see, in formulations, you obviously need a stable formulation is not always easy. And when you figure out that a solution to that problem, good for you, patent it, but demonstrate why it's an invention. And so what they did here, what the inventor did here at AstraZeneca, they were doing a pharmaceutical inhaler formulations with specific component amounts. Again, a little bit of a laundry list, and it didn't explain how any one component was the thing that made stability. But what they taught is that if you adjust all these components with optimized ratios relative to each other, that increases stability. And someone challenged it and said, look, all these components are known. Why wouldn't you just adjust them to make it more stable? Isn't that obvious? Just adjust parameters to make it more stable. But what AstraZeneca was able to show, and they built this in from the very beginning, is that no one would have thought that adjusting the relative ratio of component A to component B to component C to component D would have any effect on stability. If you knew that it could affect stability, okay, then you know to do it. But they were able to show, they found basically a new problem and a new solution to it. The problem stability was based on the relative amounts of several components, and they balanced them and they got good results. They explained this story, and they were able to preserve their patent on a commercially very important inhaler product. The next slide, please. So those are unexpected results, but you can also build your story about why what I'm doing is not what anyone else would have done. Everyone else thought this would have been a crazy idea, a bad idea, an unhelpful idea, a negative idea. I went against conventional wisdom and I showed it worked. And so this is, again, another eye drop formulation from Allergan, and they claim had the composition of 200 ppm of this BAK preservative. Everyone knew about the API. Everyone knew BAK was a preservative, and there were embodiments with pretty substantial amounts of BAK in it, 50 ppm. That was in the prior art. And the argument was, well, people knew that you could have anywhere between 0 and 1,000 ppm of preservative, and the prior art was close. You could work within that range of 0 to 1,000 and just optimize till you get your best property. Easy, the challenger said, Sandoz. What Allergan was able to show, based on evidence they built in again from the very beginning, was that what the prior art taught is they said 200 ppm going higher on BAK is bad. Going too high is going to cause safety problems. And so it's, again, the art said, don't go this way, and we went this way and it worked. And not only that, when the BAK was increased to 200 ppm, Allergan was able to show that there was not just an effect on stability, which maybe would be expected, more preservative, more stable, but they were able to show at the higher concentration of BAK, they actually increased the permeability into the eye. And that was unpredictable and unexpected. And that's the hallmark of a non-obvious invention. That's the type of information you want to develop and mine and pull and work with your patent attorney to make the absolute very most of. Next slide. We'll skip this slide. Next slide. There is a requirement that when you are going to present evidence of unexpected results, of any benefit that you might have, it has to be commensurate, is the legal term, with the full scope of the invention. So if your claim scope is very broad and covers a whole range of formulations, and you show that one formulation on the left-hand side has some special properties and unexpected properties, that's very nice, but that doesn't show that your whole claim is non-obvious. And so that example we saw with 200 ppm of BAK, the evidence they had was exactly lined up with the claim. The claim said 200 ppm of BAK. The data they had was 200 ppm of BAK. But there are examples like this case here where the claim covered a third component, and it could have been almost any amount of a third component. They had one example with a very small, or they had a few examples with a very small amount of a third component, and they didn't vary the mixture with different components, and they made it kind of harder for themselves by not showing unexpectedness across the range of the patent, and they actually lost. The patent was found invalid. Next slide, please. So we're back to another example of the same Allergan formulation. And I'd mentioned at the beginning prophetic examples. What it is this hypothetical example where they talk about, it's the text in the middle, where a drop of the formulation is administered and the pressure drops, the pressure drop is observed, lower intraocular pressure persists as long as the treatment continues. They talk about how to use it and what effects they expect to see. That supported the written description and enablement of the patent, even though they hadn't done it. And so if you're going to want to be giving, if you want to claim things, treatments of certain doses, treatments of certain durations, rather than just putting in a laundry list of numbers, it could be 50 milligrams, 100 milligrams, 1,000 milligrams. These types of prophetic examples can be very, very powerful. In the interest of time, why don't we go to the commercial success slide 2 forward. Commercial success, you can show that your invention is not obvious by saying, look how successful it was on the market. If there was so much money to be made, someone would have, and it was so obvious, someone would have made my invention a long time ago, but they didn't. And that's an additional type of evidence that you can use to develop in your favor. So let me stop here and turn it back to Dr. Dye. This is a topic we could talk about for days and weeks and 20 years, but I think we've given you an overview of some of the important initial determinations to work with and make your patents at a higher level than just a piece of paper. Dr. Dye?
Thank you so much to both Dr. Yang and Dr. Feldstein for your wonderful seminar, very educational, and we learned so much about the concepts, strategies, and also examples of how to build up a strong portfolio, patent portfolio. And also at the beginning of our seminar, hopefully the audience also learned more about Medicilon. Now I think it's time for Q&A. I wonder if we have any questions from the audience. We do have one here. Actually, I think this is for both Dr. Yang and Dr. Feldstein, right? If the claim is identified to be too broad years after the patent has been successfully filed, will the whole patent become invalid, or the company will have the chance to revise the patent to narrow down the claim to make it valid again?
Great question. I'll answer this one, if that's okay. So yeah. If you go to court and you only have one claim and it's a very broad claim and it's too broad and you're in court, you're out of luck. It's over. You don't get a chance to fix it. But the normal thing to do with pharmaceuticals, especially where they're so valuable, is that you come in and look at your patent estate before the litigation is going to start. You know the timing of when litigation is going to start based on when a generic or a biosimilar can file. And you look at your estate and you look for problems or issues like this, that the claims are too broad. And if you get it early, there are procedures where you can go back to the patent office. There's a particular one called a reissue application, and you can go back and you can fix it if you do it early. And we've done that. We've done it successfully to fix up patents in litigation to make them litigation ready. So it's a requirement to do it early. If you wait until you're before a court and they find that your one claim is too broad, you're out of luck.
Okay. Yeah. Dr. Yan, you want to add something?
Sure. Another way of just being precautious when you're filing the application is to have layers of claims to cover different scopes. You have the claim one, it's a broad one, and then you have layers of different scopes to cover from broad to narrow, such that eventually when the patent is allowed, when the broad one is gone, the narrow one still covers and is still valid. So there's another way of planning early when you're prosecuting the patent to begin with. Yeah.
Got it. Yeah, I do have some questions. So you mentioned claim early. There's a balance between claim early and also the patent life. So how can you balance this?
Yeah, so there's always a balance for everything, like the scope of the claim and when you file, right? So the proper balance is that, first of all, make sure when you get a new invention, make sure you timely manner try to file the application to get it, to avoid the future potential prior, because the earlier you file, the less prior you're dealing with. On the other hand, if you cover different aspects of the invention, that can give you very broad coverage of any other perspective invention. And the advantage of doing that is that you can sort of, if your compound patent, you file that early enough, when you're doing R&D, when the patent is allowed and expire later, you still have later filed clinical trial kind of method treatment claims still ongoing. So nobody can use your drug again. So that can make sure your product has a longer lifespan. But again, it's a balance. Why? Because if you file too many applications with overlapping subject matter, right? For example, if you have a patent covering the compound and also potential use, then when you future filing another application just patenting the method treatment, the earlier patent of yours had been published already before you file the next one, it could become prior against your own thing. So there are many considerations. That's why it takes a lot of legal expertise to sit down, and you need a good patent attorney to plan ahead and think about all the back and forths and the pros and cons and how to control the balance. Mark, anything to add?
No, I think that was a great answer. Thank you.
Okay.
Yeah, I have another question. That's
Sure.
between us. Just curious, like you usually will work with like farmers, right, or biotech. So so just curious, under what condition or circumstances you're going to work with CROs like our company? Have you done something like that?
Sure, and often it's in the context of a platform technology. And if the CRO is developing a platform technology, that we have often partnered with CROs to help them get their foundational technology that then they're going to work with their clients on. And then depending on the relationship between the CRO and its client, we often take, at one point, often the CRO data often comes through their client, and we have a mutual client basically. And we end up drafting, we have a mutual client, and we end up preparing and defending applications for our mutual client based on the work that you developed, but per your agreement, as you mentioned earlier, your client would own at the end of the day. And that's probably a little bit more common in what I see. And sometimes we have CROs that, they have capacity and it's not fully utilized, and they develop their own innovative products, and then they need to protect it for themselves and then maybe license it or sell it to a third party.
Okay. Dr. Yan, anything to add?
No, that's a good answer.
Yeah. But like,
I guess I would add one more thing. Another thing that we do with manufacturers and CROs is, in addition to the step of getting the patent, you need to make sure that you have freedom to practice for whatever you're doing. And so you may be developing manufacturing methods for some drug or some biologic, and it's very critical to consider not just is my invention patentable, but does someone else have a patent out there that's going to sue me once I start selling it on the market? And so it's very common and very complicated analysis for especially biologics to go and look, does someone else have a patent that's going to block not necessarily the whole scope, but just maybe some little part of the process, some cellular purification method, some other activation method. Is there something out there that I'm using for my own work as a CRO, for my work as a CRO on behalf of a client, where I have to make sure that we're not going to be liable and infringing on a third party? And that's a very complicated analysis to find those patents and then possibly to challenge those patents and knock them out.
Yeah.
Yeah, we have seen ongoing cases right now, between the CRO CMOs with some patent owners. The CRO CMO have their own patented or platform technology and their machinery, like the instruments. But somehow there's still some very broad patents out there potentially covering their manufacturing process. So they can sue others and they can be sued at the same time. So there are different analyses, but you have to, as Mark, you have to always watch out for your product, your process may infringe other people's broad patents.
Sure.
So we do kind of analysis to make sure you don't have this kind of a barrier, and those are analysis we routinely do for our clients. Yeah.
Right. Yeah, that's a very good reminder. So basically the CRO's purpose to serve our clients well, right? So we don't want to get make it more complicated for our clients to claim their patent. So basically we'll be the supporter, and I think it's good to know, we have strong lawyers like you guys, like Finnegan, so we know how to where to get our advice. So thanks so much. I see we have another question coming up. Should the company file several patents with some narrow patents and some broad ones, or should the company put several layers of claims, both narrow and broad ones, in one patent? Which strategy might be better? Take it.
First of all, cost-wise, filing one application is cheaper than filing multiple different applications. But otherwise, my take is usually we always see layers of claims of different scope in one patent. And I wonder, probably there are some disadvantages of filing different patents. But maybe Mark, you can tell more about from your experience.
Sure. So I think I'll just restate what you said. It's complicated, but probably the answer is both usually. That you both want within a patent, you want layers within a patent, and you want a family of patents. It's complicated because in the US, if the patent office is slow to issue your patent, and it often happens on the first patent, you can get what is called patent term adjustment, and it extends the patent beyond its normal 20-year life, and you can have a year extra protection, 2 years extra protection, half a year of protection. And in pharmaceuticals, that's where the big money is. The big money is not in year one, the big money is in year 20 when you're a big market success. And so there are things that happen though when you have multiple patents. If they're not well designed, you can have one interfere with the other and you can lose that bonus time. And so I think generally you have layers in each patent, but you'd really like to have, for example, a patent that covers the composition of matter, the molecule, the antibody first. You might like to have a second one that has a method of using it. You might like to have a third one that is a method of making it. And they're all related to each other, and they're all valuable, and they may have their own tiering within themselves. And if you set it up right from the beginning, you may be able to get all different types of families, different applications of the same invention that don't interfere with each other, that are each individually strong because they have broad claims and narrow claims, and then stronger as a family because they build that picket fence farther and farther around your territory.
And also, as I mentioned before, you can always file continuation applications. And by nature of continuation meaning you are trying to claim your further improvements of your earlier invention. So usually they're a little bit narrower than the earlier application. Usually you're talking about the scope wise is safer from the written description and enablement perspective, but watch out for the potential challenges over whether your improvement is not obvious, is inventive enough compared to your earlier invention. So they're always, you have to consider different patent law requirements when you're talking about designing the scope and the pace of your prosecution. Yeah.
Okay. I wonder, do you have any like frequently asked question during your previous presentation with somewhere else, so you can share with us?
Yes, I remember, Mark. I told you just now before the talk that when I went to China for marketing trips, every drug company asked me the same question. So, Dr. Yang, how many examples should we put in the specification to support our claim? Just tell me a magic number, how many? I always say it depends on your technology. It depends on what you're trying to claim. It depends on what technology it is and how difficult it is to teach a person art to make and use the full invention. So there's no magic number, but always the advice is as many examples and as much data as possible, because it's always a good thing to show the inventors have full possession, is entitled to the full claim scope. Mark, anything to add?
No, I think that's a great point. And I think that I mentioned it too, and you said it again. Every case is going to be different, and there's not a one size fits all, except what Yi said, almost always, having more disclosure helps you. Having more detail in your patent helps you. It gives you room to maneuver later, and it gives you further arguments. And so almost always, if you're deciding, do I include this information or do I not include this information? Almost always it helps you at the end of the day.
Yeah, and also it's not a number, it's also about diversity, right? If you disclose, as you can see, hundreds of antibodies have similar structures or sequences, the court would not care because they have to be different enough to be representative of the entire genus of the species. Yeah.
But also maybe today we're focusing on the US patent law, right? So if we're talking about the Chinese patent law, maybe there's some subtle difference in terms of number of compounds that need to be prepared, something else. I don't know if that's the case.
Yeah. I mean, I'm certainly not an expert in Chinese patent law, but my impression, certainly there are differences. And my impression is that actually getting a patent is in some cases harder in China, because you actually need better support in the application from the start on disclosing the activity and the structure that you want to claim. And in the US, you have a little more leeway than the rest of the world to sort of recombine the parts later. But it is, if you're going to do a global patent, you do need a global approach, and that's the right.
Yes, I totally agree, because my impression about my little knowledge of the Chinese patent law, I do agree. My colleagues in China, they told me the same thing. That is a little bit harder in China. It's very strict, the requirement for literal support in the specification. Again, in the US, we have a little bit wiggle room here too. For example, for a very easy and predictable field, you don't even have to provide an actual working example to show your possession of support of the claim. But for anybody, biotech is very complicated, unpredictable, then you have to show more a working example to show the real possession of complicated technology. Yeah.
So I wonder if you have one frequent last question to share, Dr. Yan? Or we're getting kind of late, especially for you and
I wanted to give Mark the closing. Mark,
oh yeah, please.
Anything you want to share?
Yeah,
Mark, you want to share something?
No, I think in terms of tips, I think we've covered it very well, and I really appreciate the questions from you, Dr. Dai, and I really appreciate the questions from the audience. I think that our contact information's available in your publicity material, and you can find E and I both at the Finnegan Law Firm website, and we'd love to interact and collaborate with any of you in the future.
Yeah, it's also our pleasure to have both of you here and to start our ACE seminar series. It's really a very good start for this year. And we are looking forward to work with both of you in the following seminar series, because you have two more, actually 2.5 more seminars coming up. It's really great. And I would like to thank Dr. Chen for the opening, and also the audience online for staying up, especially the US friends for so late. And again, Medicilon will work with everyone, including Finnegan and other clients and new clients and future clients. Let's work together and contribute to the R&D in the pharmaceutical industry. Thank you very much. Let's wrap up for today's seminar. Thanks a lot.
Thank you.
Much.
Bye, everyone.
Bye.